Oxidative?
Nitrate?
Yeah.
Because we're looking for an enzyme that reduces nitrate.
Yeah.
Right? So we need nitrate. Alright. We need nitrate. Looking for nitrate. Okay, the bacteria produces what? What is the bacteria supposed to produce? What's the assay called?
Nitrate reductase?
Nitrate reductase. Yay! If it produces this, we get what? Nitrite. Yeah. Okay. So, how do we test for nitrites? Pronounce it right, so you'll spell it right. Sulfanolic acid. Sulfanolic acid. And what? Alpha.
Alpha naphtha.
Naphthylamine. Right? And this is usually a big proponent where people get mixed up with this. Bad pronunciation leads to bad spelling. Right? And I've actually seen this happen where I've had students where all they wanted to do was ask me a question and they wrote ax. Right? So practice makes perfect. Spelling this, if you accentuate the phonetic breakdown, sulfanolic acid, alpha naphthylamine, you won't spell it wrong. Okay? Practice it because spelling counts on these, right? It's ridiculous. Okay, so we add these two reagents. What are our possible outcomes? No, what are our possible outcomes? What are we looking for? How do you tell? What are we looking for? In the test tube, what are you looking for? You add these reagents to the test tube. You did this. Color change. What are our color change?
The red.
Red and what?
And the lack of color change.
Not red. Okay. So if it's red, what does that tell us?
It binds.
We are positive for nitrate reductase. Okay, now I'm abbreviating all this stuff. You're not allowed to abbreviate it on your exam. Okay, I'm lazy right now. It's positive for nitrate reductase. How do we know? Because sulfanolic acid and alpha-methylene bind and react with NO2, which is nitrite, which produces a red color change. So we are positive for nitrate reductase. If we add these two reagents and we don't get a color change, right, that means there's no nitrite in the medium. That's all it means. That doesn't mean that nitrate wasn't reduced. So we have to add what?
Zinc.
We add zinc. Okay, we added zinc. What are our possible outcomes?
The red.
We got red and not red. Okay. So for here, right, we had no color change. That doesn't necessarily mean it's negative for nitrate reductase. So we add zinc, and we get no color change, and we get a color change. If we get a color change adding zinc, what does that mean? That means, so it reacted with what?
With the nitrate.
And don't use it, then stays what?
It reacted with the nitrate.
What is?
The zinc.
Zinc reacted with the nitrate, so we are what? Negative or positive? What?
Well, we're negative.
It was a 50-50 question. If I say try again, what does that mean?
Negative.
Negative. Right, we're negative for nitrate reductase. Why do we know we're negative for nitrate reductase? Because the zinc reduced nitrate to nitrite, which then reacts with sulfanolic acid and alpha-methylene, giving us a red color change. So because zinc did the additional reduction, we're negative for nitrate reductase. If we have no color change, what does that mean? We are positive for nitrate reductase. We are positive for nitrate reductase because we don't have nitrites, we don't have nitrates. What we have is a bacteria that reduced it so much that all we have left is ammonia and nitrogen gas. Okay? Any questions? Does this make more sense now? Because you guys are mentioning that you studied it, but it wasn't making sense.
No, it's just because I was reading it. I think I need to draw it out instead.
Okay. So, remember, you need to know these spelling counts for these two. If you haven't done it already, you have a week now, so no excuses. You need to tell me what's happening, what we're looking for, what the reagents we're adding, why we have the reagents, what color changes, what changes the reagents produce, and how we interpret those, and also what those mean, right? Red color change here means that we're positive for nitrate reductase because reagents A and B reacted with nitrite, right? It's not just red means positive for nitrate reductase. You need to tell me why. I want the complete story. This is like a 15-point question here.
So right there, if it's red, positive for nitrate reductase because they reacted with the sulfonic acid and
Yes. And also, it gets easier if you think about this in a stepwise reaction. What did you do? You added sulfonylic acid and alpha methylamine. It turned red. What happened chemically? Right? They reacted with this, producing a color change. If they reacted with this, that means this is not here. That means this was present. Okay? Right? That's a complete understanding. That way, if you see multiple choice questions that say, you know, some simple tests would say, just having a basic understanding, you add these reagents to get red. It means what? Positive A, positive B, negative C, all of the above, D, none of the above, right? Or I could ask a question that says, you add sulfonylic acid and alpha methylamine. What were you testing for? Nitrate, nitrite, ammonia, nitrogen gas, right? If you know the complete story, that's just as easy a question as telling me what red means. Okay? Understand all this. This is an essay question. If you draw it, I don't want a drawing like this. This is abbreviated. Write the words out. You have 3 hours to do this. You have enough time. If you draw out the mechanism, you need to explain each step. You can't just say red, positive for nitrate reductase, and have these up here. You need to put positive nitrate reductase because these reagents react with nitrite, proving that there's no nitrate in the media, and that nitrate reductase is present. It'll sound redundant every time you're doing this. Scientists are redundant. We repeat everything so it's thorough. Question?
And we only have 90 seconds to do this?
No. You only have 90 seconds to do the PowerPoint question.
Okay, okay.
So I will show, I'll show you an example.
I can send it to you guys if you want.
I'm so going to check YouTube tonight.
You're gonna get like 1 million views.
And I won't see a dime of it watched.
Nope.
Did your phone just cut off? I got the whole thing.
Well, yeah, I can just put it on YouTube and you guys can just look it up.
1 million hits.
Okay. So what you'll see is in PowerPoint form, you'll see this picture with like an arrow pointed here. And the question will be, this test is positive for what? A, fecal coliform. B, gram-negative. C, gram-positive. D, nitrate reduction. E, A and B, right? F, D and C. G, all of the above. That's a question. You have 90 seconds to answer that. Okay? Because usually it'll be like this. And you can actually see it, and there'll be like an arrow pointing right here to this sample. So it's not like, so in this case, right? It'll be an arrow pointing here. And it'll say, we'll just have this arrow that you can see. And the question will say, how would you interpret this? It'll be like multiple choice question. Interpret this result. The arrow is pointing to a culture that is A, positive for gram-negative, negative for gram-negative, positive for fecal coliform, negative for fecal coliform, right? It'll ask you those types of questions. So you have to be able to look at these results and interpret them. You could even see a question. I have done it before where, because you should be able to interpret the results from what we've been doing today. And the lab manual tells you too. So it's listed in the lab manual. When you do the EMB plate, it says the EMB plate is selective for gram-negative bacteria, right? So if it grows, that means it has to be gram-negative. You need to know that. I could put this plate up there and ask you what makes this media differential. A, B, C, D. Give me your answer. What makes this media differential? Say that louder.
That's not the color of the first one.
Starts with a C. hate them. Carbohydrates. Right? Yeah. Carbohydrates. Right? The sucrose and the lactose are what generate the differential. Because the ones that do this fast enough get a green shimmer, they're identified as fecal coliform bacteria. The ones that don't are still gram-negative bacteria, but they're not fecal coliform. What makes this plate selective? So that could be a multiple choice. You have 90 seconds. What makes this media selective? A, the dyes. B, the sugars. D, the fact that it has oxygen. E, Star Trek, right? Just one of those. You need to know all of those parts about these media. So I could ask, I've done ones before where we go like, bam, all the words are gone. There's an arrow pointing to this. Is this negative or positive for nitrate reductase?
Oh, with those things, would it be positive?
Yeah, exactly. You need to be able to do that in 90 seconds. Right?
I was really trying to answer. I just read it.
Yeah, so this says positive, but like you'll see something like that. Or usually sometimes I've just done the picture because we've gone over, like you add this. If there's no color change, then you add this. So I've done it with just the picture and no words before too. So you can have that. That could be a PowerPoint question. You can have something like, what else did we do? Oh. There'd be nothing down here. And I'll be like, pointing to this. This is positive for A, B, C, D. Give me your answer. So you need to be able to analyze the results. Or I could ask you, what was the limiting reagent in this? The A, glucose, B, sucrose, C, lactose, or like D, none of the above. Remember, because all the medias, you need to know the components of them. You need to know what they are, what they're used. And a lot of times what I'll get for some people, when we have other types of media, it's like, what is this media selective for? Or what's the selective agent for this? Everybody will want to put, except for the EMB plate, it'll change on the next exam. Except for the EMB plate where it's actually the dyes that do it. Some people will be like, oh, it's the pH indicator is what makes it selective. The pH indicator is never a component of selective or differential stuff. All it does is just there for color change. Okay? I could also ask you in like a short answer question where it's like, here, put it this one. What is this result telling you for microbe X? And you would say what? For like a short answer. Yes. Positive or negative for what? Positive for gas, because you have the lysine, yes. What else? We have yellow slant, yellow butt, so it has to use either lactose or sucrose, right? And or glucose. But it has to at least use two of the non-limiting reagents, right?
Yeah.
It's negative for sulfur production.
I was gonna say, because that one is like kind of pinkish red on the top, so would it be?
No, that's yellow, because this is
So
why
it's
we have
the-
control. That's
Okay,
pinkish
that
red.
must be, okay.
That's yellowish orange,
Yellow, okay.
right? Okay? But you guys have already been studying, right? So you're not freaking